thank you all for coming tonight. It's a true honor to stand here before everybody. And happy Leap Day, right? I promise there will be no kangaroo jokes. And also, if you see somebody leaping for joy in the corner, it's not about Leap Day. It's just a very joyous group that we put together here. And yeah, I can really feel it. So I'm John Assalian, and I am the Managing Director of Viewstream. And before we get started, I just wanted to thank three groups of people. So first, it's everybody that showed up tonight. Showing up is one of my core values in life, and I feel like everybody here embodies that by showing up tonight, being here, focused, listening, and really trying to understand what CRI is all about and cancer immunology research. Second, I'd like to thank Jill, Sharon, CRI, everybody involved with the CRI organization, board members, trustees, anybody that's been part of it. I really believe in the mission about bringing people together, making meaningful impact, and really creating a world immune to cancer. If you go to the CRI website, you'll see that tagline for a world immune to cancer, and very meaningful, and I think it's what kind of brings everybody together. And then just lastly, the Modernist Club, great venue, thank you for everything, and the Viewstream team, Sasha, Jeff, a lot of other people that helped organize this. Again, we've been working with them for over 5 years and just really appreciate the community, the connection, and the mission that we have. And we're just honored to be part of the purpose to accelerating cancer immunotherapy. So, any funds we raise tonight, it is a fundraiser. So any funds that you donate go directly to developing breakthrough immune therapies to treat cancer. So thank you for your generous support. And with that said, I will pass the mic to Jill.
Thank you very much, John. You're the one that I really have to thank. Without you, we wouldn't be here. Thank you for sponsoring this. Thank you for all of you here that come. I think many of you may not really know much about the Cancer Research Institute. And I'm going to give a little introduction before we get into our panels list here. But, you know, for 75 years, you know, CRI has had a persistent vision that the immune system has the capability of treating, controlling, and possibly curing cancer. That was way before the word even immunotherapy was coined, and many in the medical establishment really doubted that hypothesis. But this prescient conviction, I think, has paid off. We have been committed to supporting outstanding scientists, first doing research really just on the immune system. And once we got a little bit of a handle of how that works, then really focusing more on how the immune system interacts with cancer, a discipline that's called cancer immunology. And I'm very proud that we can really say that CRI has pioneered this field. And you know, it really, by funding the seminal discoveries upon which today's cancer immunotherapies are built, we can claim that. And we can also claim funding the scientists, many who could not get funding anywhere else, who have now become the luminaries of this field. And these leaders have said to me, with the field of cancer immunotherapy would not be where it is today without the Cancer Research Institute. So talk about impact. When I hear that, that makes me thrilled, and I know that we are doing something great. So CRI has always been, I think, ahead of the curve and tried to be a kind of visionary and forward-thinking organization. Now, while there is still a ton load of biology, I think, to still uncover before we can develop kind of the next wave of effective immunotherapies for all cancers, we come to realize that today's cutting-edge technologies, whether they're used in basic, translational, or in clinical research, are generating huge data sets that are requiring bioinformatic analysis. But I'd like to say that data on its own is not enough. You need to understand that data in a setting, in the conceptual understanding of what we already know about cancer and immunology. And so we really need to marry data science, bioinformatics, and biology together if we're really going to take, here's my leap, the next big leap into the next wave of effective immunotherapies. So CRI wants to continue to accelerate innovation and catalyze discoveries So we're evolving our programs to have a little bit more focus on data and bringing this in, not abandoning the biology and the cancer immunology, but really knowing that we have to understand how we can analyze data within that contextual setting. So we're very lucky this evening to have with us 3 CRI-funded scientists who can provide first-person views on how data analytics can deliver new insights into how the immune system works and how it can be harnessed to treat cancer. Each of them is a recipient of a CRI Lloyd J. STAR Award. STAR stands for Scientists Taking Risks. And these exceptional scientists have received this very highly competitive grant based on their track record, but also on the expectation that they have the creativity and the acumen to really connect the dots and lead us down new paths of investigation and make the next major breakthroughs in the field. So no pressure, guys, but that's what I'm saying. But I think you're going to hear when they talk that we've made some really good choices here. So I'm very proud to have them part of the CRI family. And I'll just introduce them real quickly and then we'll get on to some questions. So first I have here Alex Marson, who's the director of the Gladstone UCSF Institute of Genomic Immunology and a professor in the Department of Medicine at UCSF. We have Matt Spitzer, who's an associate professor in the Department of Head and Neck Surgery and in the Department of Microbiology and Immunology at UCSF. And Ansu Satpathy, who's an associate professor of pathology at Stanford. So thank you guys for being here. And I think before I get into kind of pointed questions, I think it's helpful for us to set the stage. So I'm going to kind of have each of you just take a little time to tell us about what research you're doing right now and how types of data that are involved in your research, why it's essential, how you analyze those data, and what you learn from them. So not too much, but there you go. Alex, take it away.
Is this working? Great. Well, first of all, thank you all for coming. And Jill, thank you for everything that you do. My lab is at UCSF and at Gladstone. We have really been inspired by the power of the immune system to treat cancer, of cells that we can infuse into patients from the immune system to go and attack cancers. And our hope is that this is just the beginning. And what we are interested in is learning the rules of the DNA code inside each one of those cells that get infused into a cancer patient so that we can go in, and now we have the tools with CRISPR to actually go in and change that DNA code to make those immune cells that will fight cancer into better immunotherapies. And we think we're just at the very earliest stages of this. You asked about data. I just want to, right before we sat down, I met Doug. Is Doug here? And Doug asked me what I did, and Doug said very quickly to me this thing that really made a big impression on me. He said, well, I have this hope that if biology can be converted to data problems, then all of a sudden it's solvable. And that is like the perfect articulation of what our aspiration is. Our goal is now with CRISPR, it's so possible to not only make one change in the genetic code, but to test hundreds of thousands, and we're getting to millions of changes. And there's a couple of billion changes in the genetic code, but with the ability to make these genetic changes, to measure the effects in immune cells of each one of those changes, and then use the latest data analysis tools, machine learning, and many other tools, it's going to be a solvable problem, Doug. And that is our aspiration, that we will understand the language of the DNA code well enough that we can rewrite it and program immunotherapies that will go after the cancers that still remain resistant to all therapies. But we have our eye on making them curable.
Thanks.
Well, I'd just like to echo Alex's thanks to Jill. It's really, CRI has been a transformative funding agency for our research program, and has brought a lot of us together to work on problems that I think are larger than any of us could have solved on our own. So in my lab, we think of the immune system as a system. It's comprised of a lot of different kinds of cells, and those cells constantly communicate with each other to make decisions, and it's a decentralized system. There's no master cell that gives instructions to everything else, but somehow this collection of cells makes choices about whether to recognize something as being foreign and to attack it, the way that we would if we were infected with, say, a virus, or to be tolerant to it so that we don't reject our own healthy tissues. And so that fundamental process is what we study. We study how these cells communicate and collectively make decisions together. And we use a lot of tools from computational tools and statistical modeling-based approaches to understand how immune cells change in their function together and provide those key communication networks in order to make that fundamental decision. And that involves taking a lot of measurements of individual cells and thinking about how those cells are changing in their functionality in different contexts and in different anatomic sites across the body to either be tolerant to a tumor and to allow it to grow and develop and spread, or to attack it and clear the tumor and provide a functional cure, which is of course the outcome that we're really trying to drive in all cancer patients.
Thanks, Ansu.
Yeah, I mean, first of all, thanks everyone for coming. It's really a pleasure to be here. Maybe I'll start with a story, which is sort of my background. So I'm a physician scientist by training. My PhD was in basic immunology, and then when I came to Stanford, 11 years ago now, thought, maybe I'm going to do something completely different. I joined a lab which was a basic genomics computational biology lab. And at the time, all of my advisors, my parents, everybody said this is a massive mistake. Immunology is just taking off. What are you going to do in genomics? You're studying fruit flies. How's this going to be helpful? And I remember sitting in that first lab meeting, I was like, oh my God, I made a horrible mistake. And then I started, trying to raise funds for my research. And I went to the NIH, and I remember I was sitting in a review panel for the NIH, and a prominent immunologist, not CRI funded, I won't say who, was like, oh, you're making all these beautiful tools, but are you actually learning anything? And I didn't get the grant. So the only person who funded me was CRI, Jill. So I think, it is, when Jill says that they're forward-looking and thinking about where the future is, not where the field has been, I think that's, I'm sort of living proof of that. And I don't think I probably would have had a lab or would have been working on cancer immunology if it wasn't for CRI. And not only did they fund my postdoc, they funded me throughout. So starting, whatever that was, 10, 12 years ago, all the way till now. Not only that, they've invested in companies that spun out that research, etc. So really full partners. Okay. So what my lab does is we think about how can we make new technologies, new genomic technologies, new computational methods, machine learning methods, to really ask the question in a completely different way, right? So not only are we thinking about the patient at the end of the day, but we're thinking about the fundamental problems, and we're trying to make these tools to really ask very basic questions about how the immune system functions, how it interacts with cancer, and how can we leverage that to make new therapies, and actually drive that cycle very quickly. The way I think about it is I think that we're trying to improve the resolution with which we can understand the immune system in cancer, and also the scale with which we can test models, test therapies, test approaches. And so you're no longer, in my PhD, it took me 5 years to test the function of one gene in one cell type. Now we can scale that 1,000,000-fold, right? So we can ask many, many more questions at the same time, and what that translates to is a more rapid timeline from basic discovery to therapeutics. So I think that's how I think about our lab broadly, is how can we build tools, methods, analysis, systems to really accelerate the process from basic discovery, bottoms up, right? Not thinking about a hypothesis in mind, just thinking about what's all the data we can gather and what's the signal we can see in that, and then take that very rapidly into a therapeutic. So as part of that, we have an academic lab where we do these discoveries, but then we also spin out many, many companies to take those forward. We're not just going to sit back and publish a paper and hope that somebody else takes that forward. We're going to do it ourselves. And I think CRI has been a partner in all of that, right? From basic biology to spinning out companies.
Thank you, Ansu. So I'm going to just ask a few questions, and then we can open it up to the audience if people have other questions. So I've heard it said that data integration represents one of the biggest bottlenecks to downstream analysis and biological innovation. So do you agree with that? And if so, what is the solutions?
I do agree with that, but I think maybe I would reframe it slightly. I would say we have this enormous amount of data, and the things that are going to determine how much we learn and whether or not it makes a difference for patients is what the quality of the data is, are we designing the experiments in ways that will actually be informative? And do we then think about how we can integrate it all and make the data available in forms that we can put it all together? So I think that we are at this moment where it's never been easier to generate data, and we finally have the ability to also make some sense of it. And as I was, Matt Ansu and I are pretty good friends too, is worth mentioning, and we work really closely together. And I think a collective goal that we all have is to use the power of data to not create data for the sake of data, but to answer biological questions that will tell us something about health and disease. And I think that we are really at that point now. The key is, can we do it? Can the biologists who are in the lab actually mixing liquids and putting things into DNA sequencers, can they be in a place where there is a communication with the people with math backgrounds and computer science backgrounds who know how to make sense of all that data? And I think CRI is clearly making a big push to bring people together from these different backgrounds to make sure that we're speaking a shared language. So it's data integration, but it's also cultural integration between all the multiple disciplines that have to come together to solve these problems.
Great answer. Anybody want to add anything to that?
Well, I think also there's been a renaissance of computational methods recently that allow us to take data that all of us may have generated in our individual labs and to think about tying it together in meaningful and informative ways. One of the challenges in biology is that every time someone does an experiment, there are a lot of factors that factor into the way that data are collected, the type of data that are generated, and other types of variables that may impact that. And it's relatively difficult to control for. But we now have better methods using tools from machine learning to actually take those data and integrate them in a meaningful way so that we can learn across larger numbers of patients. It's very expensive to do these types of experiments, and so many of us may perform experiments on 10, 20, 30 people, but it's very difficult for us to scale that to thousands of people and generate the types of large data sets that, say, folks in tech may be accustomed to working with. Even that would be very small scale for a machine learning problem that's solved in tech. And so really being able to pull data sets together, filter out the technical noise, and preserve the biological signal is a key element that I think we're making a lot of fundamental advances in, and should allow us to look now at population scales across large numbers of people.
Yeah, I think what's exciting is that all of the data that we're generating, vast majority, is from patient samples, right? So all of our labs the primary data comes from patients with cancer who are responding to immunotherapy, not responding to immunotherapy, other therapies, and you're generating data at such a scale where you can learn not only from the successes but also from the failures. And I think that's one of the lessons of immunotherapy in the field in general. That you had 30% of patients responding, getting cured potentially from PD-1 blockade, but then you had 70% that weren't. And then the field, focused on that 70% and generated as much data as we could from each of those samples that wasn't responding, and figured out, okay, here's what's going wrong in these patients, and here's how we can attack it better. So I think the data integration problem is a good thing for us. That now we're, from each of these patient tumors, blood, we're generating massive scale data on multiple modalities, right? DNA sequencing, RNA, protein, single cell resolution, and we're really able to learn a lot. So I think this is a key focus area.
I mean, I always say, I've been around so long, that we're in a place we've never been before, because there were so many years when no one responded to immunotherapies. Immunotherapies didn't work. And now that we do, it's no longer a black box. You do have comparators. You have responders and non-responders, and because of these multi-omic technologies that are allowing us to ask questions that we're never being able to ask before. So I think it is, but the amount of data is overwhelming to figure out there's a lot of noise in there, and there's a lot of what is the signal, and also how do you curate the data that's coming from, everybody's doing, you could do a 10-person experiment here, a 10-person experiment here. How do you bring that all together in a reasonable way and time frame to make sense of it?
And I think it's going back to what you said earlier. It's not going to be the data scientists that figure that out, and it's not going to be the immunologists. It's going to be the people in between, right? That have both sets of knowledge and can really make sense of what is the biological signal in a massive, and also understand technical issues and figure out what is the actual signal in this massive scale that we can attack with therapies.
So just one more question for me, and then I'll open to the audience. You did bring up machine learning a little bit, but I mean, AI is such a thing these days, and so in your day, what role does AI play for you on a daily basis? What do you do?
I'll just tell one story about this. We do experiments where in one experiment we'll take a sample of blood from a human, and then we'll use CRISPR to make 100,000 changes in the genome of the cells. So each cell will get a different change, and we will then start to learn the rules about which are the changes that might make the cells into better cancer immunotherapies. So we've published a lot of this data now, and a couple months ago I got a call from someone at Stanford. It's actually not someone with a biology background. It was someone who had previously been at Pinterest and came and was like crosstalk between tech and biology. And he said, I think I'm starting to see something interesting. I downloaded your publicly available data, and I trained an AI model on all the literature that predated your model, so independent of your model. And I asked the literature to guess which are the genes that would be useful to make immune cells that secrete more cytokines. I said, give us 100 genes. Tell me 100 genes that would be the most useful. The AI didn't know our data. It guessed 100. It gets pretty well, better than random. But then the guy from Stanford, his name is Yuri. Yuri gave our data to the AI, and he said, oh, here's how your 100 guests actually perform. We have the data. Now give me another 100 guests. And then it started to really do well. And then it said, okay, so I think we're going to see this crosstalk between data integration with AI, huge data sets, but I think we also need data curation of experiments where all the information is done in a carefully guardrailed system that provides some checks to make sure that we actually have quality control. And I think there's going to be a really interesting conversation between scale of data and careful curation going back and forth where the two can inform each other. But we need to make sure we're thinking about how to do this so that we don't go off the rails.
So we use a lot of tools from unsupervised learning every day in the lab. We make a lot of measurements about individual cells. And one of the core questions that we always have is, how are these cells related to each other, and what are the most functional states of these cells that we're trying to drive with immunotherapy? What's the end goal? And unsupervised learning provides us with a lot of tools to get at that type of question. So we can group cells together using clustering algorithms that have similar molecular phenotypes that are looking similar in protein expression or RNA expression. We use tools from dimensionality reduction in order to understand the relationship between those cells. So like fundamentally in our lab, pretty much every experiment that we do touches tools in this area in order to help us learn from the underlying biology. And I think most recently, one of the really exciting areas that Ansu's lab works on quite a bit as well is to think about how cells change over time, what's the trajectory that they're going through over time as we're trying to put them in a better and more functional state, and using those data then to identify the key signals that can make better immune cells for cancer treatment. So if we understand the path to get there, now all of a sudden we can start to identify drug targets that really help push cells in the state that we're trying to achieve. And that, I think, will be really instrumental to developing the next wave of effective immunotherapies that work in more people and provide longer-term protection against cancer.
Maybe I'll I agree with all that, and I think that there's going to be a massive explosion in biological data over the next 10 years, and I think that AI will be integral to all of the drug development process, discovery, molecule design, testing, clinical trial design, all of it. And I think you're already seeing that in pharma, and if pharmas that aren't doing that are being left behind. So that's one thing. But I think what's more interesting, and Jensen from NVIDIA said this the other day, I think what's more interesting is that now you don't actually need to be able to code to do this type of analysis and look at this data. right? So now, there was this whole wave of everybody should code and all biologists should take a computer science class, all that. I actually think that's all over now. And anybody can use a chatbot to create code and analyze data at a scale that was not possible before. And so now I think that every single biologist, every single lab will become a computational lab. So I think that it's just going to change the way that we think about, generating data, analyzing data. The mass of data that goes into discoveries will be, orders of magnitude greater. And I think what that means is that the discoveries, people say that 50% of Nature papers are never reproducible. When pharma goes and tests them out, they don't check out. And I think now if you have this massive amount of data that is underlying that discovery, I think you're going to have a greater ability to actually translate basic discoveries into therapies. So I think that would be a major.
Thanks, Lindsay. Do we have any questions from the audience?
Thank you very much for this panel. So many questions, and I'm going to limit myself. But to start off, first I want you to know that I am a cancer patient. I want you to know that because I want you to meet someone that you're helping. And I'm currently right in the middle of my second vaccine, cancer vaccine, under an FDA compassionate use expanded access application. So I've talked to lots of companies like you, they're the types of companies you talk about, and I'm sitting here right now just waiting for the Data Genetics company to send me my results to tell me if they find any more cancer cells in the blood. So, and that brings me to my question. It seems to me the problem you've got here in the US is just getting the data. Like you mentioned thousands, of patients. That's hardly enough, isn't it? I mean, if you're in a place, and I think it's because of HIPAA myself, but I've seen companies in India, China, places where they don't have those sorts of restrictions, and they can get hundreds of millions of records. I mean, wouldn't that really help your cause a lot if there were a way that you could access patient data throughout the country instead of just within your own institution?
Sure, I'll take a first pass at it and then pass it around. First of all, thank you very much for sharing that. It's really impactful for us to get to interact with people who are living the life and consumerism of the types of things that we're trying to produce. I agree with you absolutely. It would be phenomenal if we had access to larger sets of data, and I think that most patients are actually eager to have their data utilized in order to help us develop better medicines. And the next generation of medicines, even if it's on a time scale when they themselves might not benefit from it, I really do think that most people have that altruistic view and are hoping to create a future in which nobody dies from cancer and it's a manageable chronic disease or even a curable disease. I think the challenge in the US is that we do have a regulatory environment that makes that challenging, and of course we want to protect individual patients' rights to privacy to the maximal extent possible, but providing kind of opt-out systems as opposed to opt-in systems. So the default right now at a lot of our medical centers is that you have to actively seek consent from an individual patient in order to use data or material from that individual for research purposes. And there's a lot of good data and evidence suggesting that actually having an opt-out method instead of an opt-in method would allow us to capture a lot more data in a reproducible way. So I think even that kind of fundamental shift would be pretty transformative for the types of work that we do, and would still allow individuals the right to privacy when they'd like it, but also would encourage people to help us make breakthrough discoveries through having access to these larger sample sizes when they are willing to participate and would be eager to do so. So I think you're exactly right. It is a challenge for us.
Do we have another question?
Maybe I can add on to that a little bit. So I agree that having unfettered access to as many patients as possible is the right. That's where we should be going. But I think there's sort of two variables, right? One is number of patients, and the other is depth of analysis on an individual patient. And so, what is actually more impactful, having superficial data on 1 billion patients or having 1 billion data points on one patient, right? And I think that what the three of us have found recently is that, looking really deep at 3 patients who respond or don't respond to a particular therapy you could discover things that you would not have discovered if you looked at 1 billion patients superficially. So I don't know exactly what the sweet spot is, right? Depth versus breadth. But I do think that we're in an era now where we can do really deep analysis of individual patients. And I think my guess is that's probably more therapeutically relevant in terms of finding new drugs than looking superficially at every patient that gets cancer. So if I could look at your cancer or any other cancer patient's cancer, I think we could figure out things that you wouldn't figure out if you looked at 1,000 other people.
Doctors Alex, Matt, Ansu, thanks for your discussion this evening. If you had to describe to all of those in the audience that are non-scientists what 3 diseases, either immunotherapies or cancer-related, you think are going to be impacted in the next 5 years, based on your basic research, but the basic research around the world, what are the top 3 diseases you would consider?
It's a great question, and it's always a hard one. You know, we always try to balance optimism with reality. But I'm going to give actually just a very blunt answer. One of my colleagues over there works at a company that I helped to co-found that I'm incredibly proud of, called Arsenal, this is a company that came out of collaborative work that was supported in my lab by CRI, supported in Cole Roybel's lab by CRI, a number of labs have contributed technology, and Arsenal has taken this to the next level. And so I'll just give the practical answer of what we're trying to do at Arsenal. Arsenal is now in clinical trials for patients with incurable forms of ovarian cancer that have been refractory to all chemotherapies. And we just opened up a second clinical trial that's going to start to enroll patients with kidney cancer. So we're holding our breath and waiting for the results, but that's where we're starting, and we hope that it continues to march out there through solid tumors that have been resistant to cell therapies for cancer.
Well, adding on to that list, in my lab we largely focus on breast cancers and head and neck cancers. Those are the 2 main types that we study in patients, and I'm really optimistic about the outlook for both of those classes of individuals. I mean, breast cancer is the most commonly diagnosed cancer in women still, and currently there's a glimmer of hope that there's a fraction of patients with a subset of breast cancer that respond really well to currently existing immunotherapies, but unfortunately most patients don't. I think we have a pretty good handle on what's going wrong in that setting, some mechanisms of what we call resistance, of why the drug isn't working effectively. And we're now working on strategies to reverse that. In the lab, we're seeing really good results in mouse models and translating that, hopefully into patient settings here in the near term. So we're really optimistic on that front. And I'll pass it on to Ansu.
I'm not going to answer your question. I think that what's really exciting about immunotherapy is that it's broadly applicable across multiple cancer types. So even though we might work on one cancer, I think PD-1, the first trials were done in skin cancer, right? Melanoma. Now PD-1 is approved in a dozen other indications, right? Because the immune cells, the pathways, the molecules, they're all the same, right, in every cancer type. So I think what's exciting for me is that it's this concept of machine learning, transfer learning. What you learn in one setting is highly applicable to another setting. And you might be surprised where that actually takes you.
Thank you. Any other questions? Yes.
Thank you so much. I'm Yash Banagar. I work as a life science investor at a fund called Nova Holdings. So really appreciate the discussion. One thing that I've observed is that we have so many exciting new modalities, and immunotherapies, CAR T, bispecifics, NK cell engagers. None of them are perfect. Everyone has room for improvement, and I'm curious how you all think, if you look at the existing toolbox and you look 5 or 10 years down the line, how do you see them improving and becoming more accessible and efficacious and safe for patients?
So, nice to meet you. I think I'm coming to your office and so I actually think this is where data can be quite helpful, right? Because you mentioned BiTEs and ADCs and CAR-T. A lot of where one works and the other doesn't has to do with the target. Is it expressed highly? Is it internalized? And if you could actually measure that in massive scale of data, you would actually prioritize the right modality for the right target, for the right cancer, for the right patient, right? And so I actually think this is exactly the area where data would be translationally helpful.
Anybody else?
I guess I don't have an answer about what's coming up. We're all pushing, looking for more modalities. I guess I just feel incredibly grateful to, I already said we're at a moment where we can generate more data by orders of magnitude than ever before. We can use machine learning to interpret it. But the third part is that we can actually do something about it. And to have these growing set of modalities, we can actually take genetic information and intervene on it. And whether that's coming up with new checkpoints that we make biologics against, whether we discover new targets that we make CAR T cells or BiTEs against, or finding antigens that we can make into vaccines, and that now we can deliver with lipid nanoparticles. We just have so many different points of intervention that if you can make biology into a data problem, you can not only solve the problem, but you can then bring it in with realized modalities and actually do something about it.
Anybody else? Yes.
I was curious if any of you use organ chips in place of animal testing, or if you're looking at that, or that always
Organs on a chip.
Yeah.
We don't, but not that might change any minute. We are really committed in my lab to trying to do everything that we can do in human cells, and we want to try to do our best. And this is not perfect. There's times when we have to use other models of disease, but we want to be able to actually look at the effect of the human immune system in human immune cells. And that means that if we want to see how those interact with cancer, we need to find better ways of modeling the cancer. And so we're thinking about what the right answer of how we model those interactions between the immune system are. But the real answer is that we have to get into clinical trials. But we need to be prioritizing how we get there. So we haven't quite got into organ on a chip, but there are people in this audience who I know are working on organ on a chip, and we're thinking a lot about organoids of different parts of the immune system, cancer organoids. And so I don't have a perfect answer, but it's another area. We need progress, but I think we're also seeing progress.
In our lab, CRI has actually given us funding to start working on these types of systems, which we're really excited about. One of the things that we spend a lot of time thinking about is where immune responses are initiated, which are in lymph nodes. Lymph nodes are little structures that are all across your body, and they look at material that's coming from every organ and see if there's something foreign there that you should mount an immune response to. And you may be familiar with your lymph nodes swelling after you get a vaccine, or in your neck when you're sick with a respiratory infection, for instance. So this is where T cells get activated. And CRI gave us a grant to work on making organoids from lymph nodes of cancer patients so that we can stimulate those with drugs and see which drugs the cells are responding to. And it's working really well now. We're really excited about it. And again, it's one of these projects that was so pie in the sky, the NIH would have never given us money to do it, but CRI did. And I'm so grateful for that.
I love the plugs. We all like hearing how well good CRI is doing. So I think that with that, I think we'll end, and we're still around, and I'm sure people will ask questions, but I think we can have another drink and have some more food. And thank you again, John, for doing this. So thank you all for doing this.